Thank you so much. Thank you very much, and sorry for all these delays. So I'm very excited to be here, and I was actually changing my talk this morning, listening to all the amazing talks that there were in the session this morning. And so we've heard a lot about going from DNA to thinking about DNA sequence information to thinking about gene expression. And then we had one talk in the morning by Sean, which actually went to also epigenetic marks, but again, epigenetic marks in 1D. And thinking how that actually has an impact on gene expression. In particular, we saw really nice examples of transcription factors, which will actually give rise to very different gene expression patterns, dependent on the epigenetic marks that you actually have. And so I thought that since this morning we've never seen anything about the 3D organization of the genome and how important that is for gene regulation, I thought I'll actually concentrate on that kind of mechanism for gene regulation here in this talk. And so I've been very, very lucky, and my group has been very lucky to work over 7 years very closely with G.V. Shivashankar's lab at ETH Zurich, who's an experimental biologist. And he has really, changed kind of the paradigm of how one is thinking about this mechanochemical coupling between cell shape and gene regulation. And now I'll say a little bit about how that relates to 3D genome organization and why that shows the important role of 3D genome organization. So he has done these really amazing experiments where you take the same cell, so everything is the same, and all you're doing is you're just plating it onto different geometries. And these cells, so same cell, same signals, you can also do it without any signals, just plated onto different geometries, actually give rise to very different gene expression programs, so gene expression outputs. So that just already tells you how important the 3D shape is of the cell and of the cell nucleus. Okay, so that's the only thing that has changed here. So you have these same cells just plated onto different geometries. They actually give rise to very different gene expression programs or gene expression outputs. And these are really predictable. So you can in fact predict these as well. Now, he then proposed, and this has led to a lot of the computational work in my lab, he proposed that, that the really important intermediary is the 3D genome organization. So if you change the shape of these cells, and therefore also change the shape of the nucleus, you actually change the 3D genome organization inside the cell nucleus. And so if you have different genes that come close in space, well, you can have the same transcription factor that goes in, but it will actually turn on different genes at the same time. So you'll get very different co-regulation patterns, very different gene expression patterns that come out from that. And so that has motivated a lot of our work together of trying to actually come up with a computational framework of thinking of gene regulation, where this 3D chromatin organization is this intermediary that is really important to understand how mechanical constraints on cells actually then can give rise to very different gene expression programs. And this is really causal. I mean, I should also point out some really amazing work by Shiva's lab on the experimental side, during the last couple of years, showing that you can actually even reprogram cells without any external transcription factors. So no addition of any chemical factors, just changing mechanically the shape of cells. Okay, so over prolonged periods, if you just change the shape of cells, you let them divide on these changed shapes of cells, you actually can reprogram cells. So you can, for example, go from fibroblasts to induced pluripotent stem cells. Okay, so that tells you that this is actually causal, right? So this is a really, really important kind of mechanism or input that we really have to take into account, and that I think is not really taken into account very often when you think of RNA-seq data, because there you don't get an image to tell you also about, in what kind of mechanical state these cells are. So a lot of the variability that you see in single-cell RNA-seq can actually be attributed to just different mechanical states of these cells. So it's really important to keep that always in mind. And just to show you how much information is actually in the 3D organization of the chromatin, and again, because this morning we saw everything always on sequencing data, I thought I'll now just talk about imaging data. So the cheapest way to measure chromatin organization is just using DAPI. Okay. So I just use a fluorescent label for the DNA, here, for example, DAPI. And I'm here showing you that just DAPI-stained cell nuclei can very easily distinguish here, for example, between different states in breast cancer. And now if you're a pathologist, you already know that the nucleus shape is very indicative of which state you are. But here what I'm showing is that even if you take just a little, patch inside the cell nucleus, this contains no information about cell shape. Even this, like, you can very easily tell, and this is like same cell type again, just all we're giving it is just a small TNF alpha signal. You can very, very easily distinguish between the cells that did get the TNF alpha signal and the ones that didn't. Okay, just telling you how much information is actually in the chromatin organization, just captured by a very simple modality, which is DNA staining. So just these DAPI stained cell nuclei. Okay, so that's kind of will set the framework for how I'm going to think about it and how I'm going to relate this all to gene regulation. Now let's think a little bit more about on the machine learning side of what are the tools that I want to use. So first, I think also thinking about this morning where we saw a lot of machine learning tools that have been developed in machine learning introduced for other problems introduced into biology. I want to just that we should always be aware of the fact that all these machine learning models were introduced for problems that were not biology. Okay, so in particular, the key foundational developments in machine learning today, they have been introduced motivated by recommender systems, by online advertising, and by self-driving cars. So these have been like the three main motivations for all of the foundations in machine learning that have been developed. And these are the tools that we saw applied this morning. Now, I think it is important to note that in biology, we often have a different end goal than in these particular applications that have motivated much of the foundations of machine learning that we have seen until now. In biology, the ultimate goal is often to actually understand the underlying mechanisms, to get a causality, to be able to predict the effect of an unseen intervention. So that's a very different goal than kind of in, say, recommender systems where you really don't care. You're not going to make a lot of money by understanding the causal, what drives, this in a recommender system, for example, if someone buys sunscreen, why this person might also be interested in ice cream. That's not how you make money, right? All you have to do is be able to predict that these two things are correlated. It doesn't matter that, the underlying causal relationships would be that, now it's sunshine and it's warm, etc. And therefore, a person that is interested in sunscreen might also be interested in ice cream. Okay, so there's very different kinds of end goals that you have in biology as compared, or many problems in biology as compared to, in the kinds of applications that have motivated a lot of the foundations in machine learning today. And so I think it is very important to always keep this in mind. And I really believe that we need to change what machine learning is today, and to motivate it, to motivate what the machine learning tools that we're developing by actually the underlying biological questions that we're asking. And so here in particular, what I'm going to concentrate on, I first wanted to just concentrate on the possibility of, or the availability of a lot of perturbational data. What I'm now here going to concentrate on, given the talks this morning, is maybe more about the availability of a lot of multi-view or multimodal data in biology, be it sequencing data that we saw a lot this morning, but also a lot of imaging data that is very cheaply available, that we should, understand how to best integrate it and what can we actually gain from having these multimodal views. Okay, so what are the types of models that I will be using? And then how do we innovate them in order to actually get to, having models that are motivated by the particular biological questions that we want to study here. So, I don't really need to go into what are autoencoders, but what we will be using. So first of all, representation learning is critical, as we've already seen in this particular picture here. And throughout the talk, there are, of course, a lot of different models for generative modeling. I will be using autoencoders just because we have developed so much of the theory to actually understand when they work, under which conditions they work, how we should use them in order to make them work for the particular biological questions that we want to study. Okay, so these are autoencoders where you have a neural network that comes into the latent space, another neural network that goes out to the latent space, and you're trying to minimize reconstruction error here. And in particular, because we will want to study, the use or the integration of multiple different modalities, you can actually do multimodal learning using autoencoders. So we have different modalities. Think of this as single-cell imaging, this as single-cell RNA-seq data, this of, say, single-cell ATAC-seq, single-cell Hi-C, whatever you have access to. And you can train one autoencoder per modality, and then we're going to glue them together in the latent space by just training another classifier in the latent space that you want to fool. Okay, so you want to not be able to tell whether a cell comes from a TAC-seq land or imaging land or RNA-seq land, right? So I want to have an integrated representation in here. And so that means I can even do this with unpaired data. So single-cell RNA-seq and single-cell DAPI cannot be obtained in the same cells. Okay, so I take some bag of cells, I take out some for imaging and some for sequencing. They come from the same distribution. So in the latent space, I should not be able to tell whether a cell came from imaging land or from RNA-seq land. So I'll just add a classifier to make sure that the distributions are actually matching. Okay, so that's how you can do multi-domain translation. And, you know, these similar ideas have, of course, been used in computer vision a lot of, like, you know, I have some brown-haired women, black-haired women, blonde-haired women, brown-haired men, etc. And I take a, say here, a brown-haired woman, and I want to predict or generate the image of how she would look like were she a blonde-haired man, for example. That's how you can go from, you know, translating between very different data modalities. And we, in fact, will do and have introduced, you know, as one of the first groups here, has introduced in the single-cell biology a framework like this, where we show that we can actually go from chromatin images, which are, of course, very, very cheap data modalities to obtain. We can predict the corresponding RNA-seq profile. So this is literally going single-cell from single-cell chromatin image to predicting the single-cell RNA-seq profile. Now, the question is it just a standard autoencoder that can do these kinds of things? And how do we actually have to, you know, update the current machine learning methods in order to do something like this? Okay, and I'll show you validation of this stuff later on, which we can nowadays validate very easily in spatial transcriptomics data, where we do have access to imaging and then also transcriptomic information in a paired way. So we can literally validate that we can actually do this. We can take cheap modalities and really translate to these single-cell RNA-seq modalities without any issues, in fact. So that means that these images contain as much information to be able to predict just the RNA-seq profile. Okay, so what are the kinds of innovations that we need on the methodological side, or that we have introduced? Okay, so one is over-parameterization. Okay, so autoencoders standardly are like PCA, right? Where you go to a lower dimensional representation, you want to go to this lower dimensional latent space. Now, what we're doing is, in fact, we're not using autoencoders in this way. So we always go to a higher dimensional latent space than what we started off with. Okay, so this will sound crazy. Okay, so you start and say 20,000 dimensions, you go to 50,000 dimensions. Okay, so why should you ever do something like this? Okay, so it has never been introduced because it literally sounds crazy. But, you know, in particular, it sounds crazy because now I don't have a bottleneck, so I could just learn the identity map. But I'll prove that you're not learning the identity map and that it's actually doing something useful. Now, where does even the intuition come from doing something this crazy? Is that we have, you know, over recent years, when have neural networks in the classification setting become useful is when they became over-parameterized, okay? When they had more parameters than the data sets that you're actually putting in. So why not apply the same kind of thinking also in the autoencoder setting, okay? So the neural networks nowadays, the ones that are successful, are always trained to zero training error. Right? And so the question is, why do they then generalize if they're trained to zero training error? So there is a whole theory now in machine learning of why that is the case, right? So you can get to zero training error and you can at the same time still generalize. And in fact, we proved that, you know, in the classification setting, we actually provided a full story of, like, if you let your neural networks become infinitely wide, what are the functions that are being learned and what are the optimal types of activation functions that you should be using. Okay, so in that setting, you know, we know that the optimal activation functions are in fact not your standard ReLU, they're not sigmoid, there are none of these kinds of activation functions that people have generally used, but they are in fact very simple polynomials that you can just use in your neural networks that are provably optimal. Okay, so this gives rise to the classifier that is an optimal classifier. It's known as a Bayes optimal classifier. So we understand completely, if you go to very, very large networks, you know, when do they actually give you the optimal classifier that you can possibly get? And so all of this kind of motivates that we should also do something similar in the autoencoder setting. Okay, so what are the functions that are being learned in these crazy autoencoders? So again, we're just using autoencoders to embed into an even larger dimensional latent space than what we started off with. So this is how you should think of it. So we have an input layer, and then we have this latent space that is larger dimensional than what you put in. Okay. Now, what are the functions that are, how can we even test what are the functions that are being learned? And why is it not the identity map? So it's not, so there are papers that suggest that it is the identity map, but we have a proof that it is not, and you can, for yourself, prove that it is not very easily. So just train such an autoencoder, put in a test image. So here we're just putting in an image, which was a training example, and we add in a whole lot of noise on top of it. Okay, now an autoencoder is pretty nice, right? Because whatever comes out, I can put it back in because it has the same dimension. Okay, so I can just, do an autoencoder, map my image through it and put it back in, whatever I got out. And so if it was the identity map, it shouldn't move, right? So just do this many, many times. And what you'll see is that it actually converges to one of your training examples. And this is provable. Okay, so whatever you put in, it will always come out one of the training examples. So this means that these autoencoders are actually learning contractive maps, and they are contractive at the training examples. Now, why is that a good thing? Okay, so, memorization would be if it would immediately give you out one of your training examples. That's not what it does, okay? So you do have to iterate it many times until one of your training examples comes out. Now, this is a good thing because, right, so you would want a map, so an autoencoder that is that should generalize, should learn a map that if you put in something that is close to a training example, it shouldn't map you to something that is very, very far away. Right, that would be a crazy function to learn. So you definitely want something that when you're close to a training example, well, it should either move you a little bit closer or at least at the same distance, but certainly not to something like very, very far away, because you're pretty certain about how the manifold looks like when you're close to your training examples. And that's exactly what these over-parameterized autoencoders do for you, just for free. No added loss functions, blah, blah, nothing like that. Also, they're much easier to train when you're actually going to these over-parameterized latent spaces. Okay, so first of all, that's the one thing that we're doing, is always using these over-parameterized autoencoders. The next thing is, how does multimodality help you? Okay, so what I said is, like, we're adding single-cell imaging data and single-cell transcriptomic data into the same latent space. Now, here's how it relates to causality. And in fact, only this year were we able to prove that the intuition that we had all along when we were doing this is in fact true. At least in the linear setting, we can prove it. So, this was always the intuition that we had, that, these multimodal representations, they will give us something that is closer to causal than if we would use an autoencoder just in each modality by itself. Because what is a causal feature? A causal feature in a cell? A causal feature should be there no matter how you're looking at the cell. Whether you look at it from left or from right, from modality A, from modality B, from modality C, whatever is causal should always be there. So it should be invariant to how you're looking at the system. So it should be something that is invariant to whether I'm looking at it from imaging land, or I'm looking at it from RNA-seq land, or from ATAC-seq land. And so that's exactly what is represented in this latent space, in this joint latent space. So this is invariant to all of the different data modalities of how I'm looking at it. Okay. So here, this means that representation learning could be used as a tool for causal feature discovery by learning these integrated latent spaces. Because causal features should be invariant to how you're actually looking at the cell itself, right? It should be something that is always there as a causal feature. And in fact, we now do have a proof. I won't go through it, but you can find this paper on the archive. We also have similar proofs that were just came out in ICML, actually presenting, I guess, this week, where at least if the causal model is linear, then we can prove that, in fact, with different data modalities, if you're learning these joint latent spaces, you can provably identify what the latent causal features are. Okay. So these are causal features that I'm not giving you what these features are, and I can actually do causal learning of what are the causal variables just by doing multimodal data integration. So all of this intuition of why I should be using multimodality of, like, imaging and single cell RNA-seq into a joint latent space is actually here has like a theoretical underpinning that it does give rise to causal features. And we believe that these theorems extend also in the nonlinear setting. We just don't have the theorems yet. Okay, so that's the intuition for why we should be doing, first of all, why we should be using over-parameterized autoencoders. Second of all, why we should always be integrating them into a joint latent space, because that gets us much closer to causality than, if you would just use your standard autoencoders. Okay, so now what I want to do is just end with, one story where I go a little bit deeper into how we're actually adding in all of the methodology that I just showed you in the spatial transcriptomics context. Okay, and how does overparameterization help you? How does multimodality help you, etc. Okay, so this is the data set. So we worked together with Xiao Wang at the Broad, who developed StarMap. And so this is a StarMap data set in Alzheimer's. So what does StarMap data give you? So it's a spatial transcriptomics technology, imaging based, where, you know, what you see here, what is nice is it gives you chromatin images. And it gives you gene expression. So here in this particular data set, this StarMap version had over 2,000 genes. So you can think of it as, you know, this cell by gene matrix that you get out. So you know where the cells are here. And so you can measure all of the gene expressions. And also, because it's spatial transcriptomics, you also get the adjacency of each cell. Okay, so I get the 3D location of a cell. I get its gene expression output, a 2,000 dimensional vector. And I also get its chromatin image. Okay, that's what you get in StarMap. In addition, this was Alzheimer's, so what she also fluorescently labeled is plaque. She also had one more fluorescent label that we didn't use, tau tangle, but we're going to use plaque here. So now this is our multimodal. Now that we've gone through the theory, all we're introducing here is exactly what I described. An over-parameterized autoencoder. You see here we're going to a larger dimensional latent space, and we're going to now put together 3 modalities. Okay, so gene expression goes in, adjacency goes in. Adjacency can go in using a graph neural network. So that's the other nice thing, right? When you have these autoencoders all going into a joint latent space, I can use different kinds of autoencoders or different kinds of architectures for every modality. So for the imaging modality, I'll use a convolutional neural network. Here, I'll just use a standard fully connected neural network on gene expression. On adjacency, I'll use a graph neural network that can actually take into account the structure, the 3D structure here of, you know, which cell is a neighbor of which other cell. What is also important when you're doing this is always to reconstruct all of the modalities. Okay, so there have been quite a few works now that don't reconstruct adjacency, for example. They don't reconstruct imaging. So you're actually getting a lot out of really trying to, you know, decode all of the different modalities. So what does this mean in the latent space? So we're all familiar with, you know, just standard autoencoders in RNA-seq, right? Their 2 cells will be close if their RNA-seq profiles are close. Now in this one here, where you're adding all 3 modalities, 2 cells in the latent space will be close if they're similar in gene expression, if they're similar in chromatin image, and if they have similar neighbors. Okay, that's what this model does. So it will find embeddings where 2 cells in the latent space are close if they have similar gene expression programs, similar adjacencies, and similar chromatin image. Okay. So now let me just show how this works. So here in this particular data set, we have 4 mice, 4 months and 8 months old. 2 are have Alzheimer's or in the Alzheimer's model, and 2 are normal. And so this is, so we map all of the cells, right, are 1 dot in the latent space. Then we do clustering, like you standardly do with RNA-seq. We do clustering in the latent space. So these are all the clusters that we got out. And then we map every cell back to where it came from in the mouse. Okay, so this red dot here is just, you know, this cell here, it mapped back to this position in the mouse. Okay, so these are just the clusters mapped back to these brain slices in the mouse. And you see here that they're very nicely consistent. I didn't have to do any batch correction. So overparameterization actually takes care of that. So I didn't have to do any batch correction here at all. These clusters all are consistent across the different mice, although the slices are always slightly differently. And here you see what happens when you use your standard autoencoder. So standard autoencoder has huge batch effects. So for example, these cells here are not clustered together with these cells in the other mouse. Okay, so across mice, you get clusters that are completely inconsistent with each other. So that's just something that comes out directly of overparameterization. Now, this is not magic. I mean, we have, you can check in the data set whether, in fact, for your data set, overparameterization will literally just get rid of the batch effects. So that's something that is quite nice that you can actually do here. So you don't need to use any other loss functions in order to get rid of your batch effects. So that's one thing. So that's how overparameterization helps. So now let's look at different downstream tasks that you may want to do. So one downstream task is something that I already talked about before, Well, here it is because I'm measuring, or because with Xiao's really amazing technology, she can measure transcriptomic data and chromatin imaging in the same slice. We can actually validate that our translations work, right? Because we get it for the same cells. So what we did, and that's task 1, okay, is just to literally show again that we can go directly from chromatin images to gene expression. So what we did is, so these are again our 4, oh, sorry, I got the months wrong. So these are our 4 mice that we have. And we completely left out the 8 months control mouse. Okay, it was not used for model building. It has never been seen. And so now we're doing, that's how we trained our autoencoder. Now we're adding in this 8 months control mouse where, this is your, and now we just do our standard RNA-seq analysis, what people do, okay? So you embed your RNA-seq data of the 8 mouse of this data set that you've never seen. You do your clustering, okay? I do my clustering in the latent space, and then I try to figure out what are my genes, my marker genes for each one of the cluster. And you see here that, the clusters that we get out on this 8 months mouse, we have never trained on it, are very consistent with the clusters that you would get out. So again, note that this was just going, so we take 8 months mouse, we only give it the chromatin images, we're generating the RNA-seq profile. Now on the generated RNA-seq data, we're doing clustering. And we're getting out the marker genes. So this was all based on chromatin images. We get out the same clusters, basically DG, these excitatory neurons, and we get out the right gene markers here as well. So just based on chromatin images. So that's task 1. So just to show again that you can actually translate between a very cheap modality to this gene expression modality. But let's do something a little bit more meaningful. Which is, actually trying to do something that is disease relevant. Okay, so now we have chromatin images and we have single cell RNA-seq. Let's see whether we can get better biomarkers of disease when we have these integrated latent spaces as compared to just one of the latent spaces. Okay, so we have these integrated latent spaces. And now what we're trying to predict, so as I said, in this particular data set, we also have plaque, right? So now what we're going to do is, based on the embedding of a cell, we want to predict whether there is plaque nearby and how much plaque there is nearby. Very simple prediction task. Okay, so I have all my cells in the latent space, and all I'm doing is training here a regression model that predicts whether and how much plaque I have close by. That's all, okay? And then I can look back at what are the features that are being used when you're doing this prediction task of seeing whether a cell has plaque nearby or not. And now we're going to look at this in the imaging land because, generally people are more comfortable with looking at it in expression land, where we know there are, for example, microglia that have particular expression programs that, when they're disease associated, so close to plaque, these are the particular expression programs that show up. So let's look at imaging land. And so here what you see, what is highlighted, is actually pixels that are very indicative of having plaque nearby. Okay, and you see, first of all, that cells are not completely highlighted or not highlighted. Only particular parts in a cell or here in a nucleus are highlighted. And this again comes from the biological collaborators who looks at this and identifies that what is actually highlighted is heterochromatin in the cells. Okay, so, wouldn't have been clear to me, but okay, so if heterochromatin is highlighted and that is the suggestion, well, then let us just plot heterochromatin to euchromatin ratio. Okay, so that's all. We're just plotting heterochromatin to euchromatin ratios based on the images themselves. And in fact, looking at how that relates to the gradient of this, in this case, a regression function that is learned. And indeed, these heterochromatin to euchromatin. So what the neural network is really picking up is something like heterochromatin to euchromatin ratio, which is quite nice. Okay, so this is really what it is using in order to predict whether there is plaque nearby or not. And this is what is also really nice about it is once you have these features, you can actually get much better gene expression markers. So generally how people pick up what are the markers of disease-associated microglia is they just take all of the microglia that are close to plaque and they do a differential expression analysis of these microglia as compared to microglia that are further away. Now, here you will see that not every microglia that is close to plaque is actually indicative of the disease. So you can get much better markers when you remove these microglia like this one here, that is not indicative of the disease, and only take the ones that the model actually identifies as being disease associated. Okay, so in this way, you also get much better markers when you're using both of the different data modalities together. And what was exciting and has not been known is that, in fact, basically all cell types, so neuronal and non-neuronal cell types, all show this tendency that if you're close to plaque, or if you are in an Alzheimer's disease model, you actually have more heterochromatin as compared to euchromatin. Okay, so this is a trend that is actually, so disease association is shown by a larger heterochromatin to euchromatin ratio in all of the different cell types. And so I will end with this and just end by saying that, cell biology, I think, is really uniquely suited, not only being one of the greatest beneficiaries of research in machine learning, but in fact, one of the greatest sources of inspiration for it. We should really build new machine learning models that are developed, whose inductive biases are aligned with the biological questions at hand. And therefore, what it means is we do need representations that are causal. For example, they should be able to predict the effect of unseen perturbations, which these representations can. I just didn't go into it, but I do have a reference here, for example, if you want it. And I wanted to highlight how much information is actually in the 3D genome organization, even just represented by cheap chromatin images, not even using high-C data, etc., but just these very cheap chromatin images already contain all this information. And with this, I will end. And sorry again for all the difficulties at the beginning.
Well, thank you very much, Colleen. So please raise your arm.
Before you start, everyone, just quick announcement. We set up row 4 as an overflow room. I think that just what came from there. It's also pretty full, but we have a second room. You can try to make it in there. There might be some chairs and some space. Sorry, it's the best we have at the moment.
All right, so we can have 2 questions.
Hello, thanks for the fascinating talk. I have a question about the causal learning algorithm that you presented briefly. So when you say you're learning the latent causal vector, how does it relate to like many causal discovery algorithm requires you to observe all the variables that are involved in the mechanism?
Correct. So here, not at all, right? So what was the main motivation is you want to do causality based on images. Okay, so generally what you assume is that you know what the causal variables are. So you get RNA-seq data. I know that my nodes are the genes and I want to learn a causal graph among them. But in an image, what are the causal variables, right? You don't know. Is it like how much transcription factors in the cell nucleus? Is it the shape of the nucleus? Is it, anything else? So I need to learn what the causal variables are. So they're latent. So that's what this does. It actually learns what the causal features are, and then it learns the causal graph among it. Right? So this is, I think, what the ultimate goal should be in causality is it's not true that we know what the causal, we just, because we measure gene expression on genes, we assume that the right way of looking at it is at the level of genes. Not clear. It could be, pathways, etc. So this is what this is learning. Yeah, great question. Thank you.
Yeah, thanks for the talk. That was great. I kind of have 2 questions, but they're really quick. With the giant latent space as well, when you're clustered on that, because it's like an over-parameterized latent space, is that actually taking 2 specific components from that space, or is it's just a t-SNE of the space itself? Or
so we're not doing clustering in the t-SNE, right? You're doing clustering in actually the large dimensional latent space.
Okay, yep. Yeah, and is it just like a
standard? Here we did ligand clustering.
Okay, fair enough. Yeah, and then the other one was just about the kind of modality and variant causality. Just that idea as well. Would you think, are there any like cellular features that would maybe fall over for, like just theoretically, I suppose that, the measurement in one modality of a feature is going to not speak to the causal nature of that feature, so you're not actually going to see the same thing?
Yeah, so actually our theorems are already in the setting, which has a partially, so one part, as you see here, only some of the features are overlapping between all modalities, and we're allowing it to have latent features that are only modality specific, because we do believe in biology that there will be some features that you can only measure in imaging land or sequencing land, etc. so we have the theory for it. But when we did this work here, we did assume a fully shared latent space. But you're completely right. Yeah.
So there's one online question that I quickly want to read. Like, how important is it for the model training that all modalities come from the same sample, so it's like matched, and would prediction performance drop by a lot?
So this is not matched, right? So this is the nice thing about this framework, is that it can be used in an unmatched setting. In particular, here it was not matched. You cannot measure DAPI and single cell RNA-seq in the same cell. But we did validate it in the match setting where we do know what it should be. So we did validate it here already with ChIP-seq, RNA-seq, ATAC-seq. And then, of course, in the spatial setting, it's an easier problem because it is matched. But the theory, nothing requires that, which is nice. But for validation, it's, of course, great.
Okay, good. Well, let's thank Caroline.